So usually when you think about the most lethal infectious diseases in human history, you probably picture these hyper-aggressive, unstoppable, biological villains.
Right, yeah, like something fundamentally evil at a microscopic level.
Exactly. But what if one of the fastest killing diseases known to humanity? I mean, an illness that can drain the life from a perfectly healthy adult in a single day is caused by a microorganism that is, for the most part, completely harmless.
It really is sort of the ultimate biological paradox. Because, we tend to assume a deadly pathogen is born deadly, that its entire existence is geared toward invading a host.
Right, like it's designed to hurt us.
Exactly. But with this specific organism, we're actually looking at something that is naturally peaceful. It's just floating around in coastal waters, completely minding its own business for most of its existence.
And that paradox is exactly why we're diving into this whole stack of microbiological research and epidemiological files today. So welcome to the deep dive, everyone.
Glad to be here.
Our mission today is to unpack Vibrio cholera. That is the bacterium responsible for epidemic cholera. We are going to explore its microscopic machinery, the bitter century-long fuse over its discovery, and, well, why this disease is actually still a live global health emergency today.
Yeah, and the overarching theme you'll see emerging from these sources is that cholera is really a masterclass in how a microbe interacts with its environment.
This means we can't just look at the germ itself.
Right. It forces us to realize that human infrastructure, like our pipes, our water treatment, conflict zones, that is the ultimate decider of life or death here. The bacterium is simply a trigger in a much larger, ecological and societal chain reaction.
Okay, so to understand that chain reaction, we have to kind of completely upend what you might assume this organism actually is.
Oh, absolutely.
So the baseline microbe, Vibrio cholerae, is this comma-shaped gram-negative bacterium. And the sources point out that its gram-negative structure means it has this really robust outer membrane.
Which is crucial, yeah, because it helps it survive in its natural habitat.
Right. And that natural habitat is not the human gut. It lives in brackish estuarine water, basically, where rivers meet the sea.
Yeah, and it has this single polar flagellum. It's like a little microscopic outboard motor that propels it through the water. But in its default state, it's just a water bug.
Just a harmless little bug.
Totally. You could sample coastal waters all over the globe, from the Ganges Delta to the U.S. Gulf Coast, and you would find naturally occurring V. cholerae.
But they don't make you sick.
The vast majority of them. No. They'll never cause human disease. They just sort of attach to the shells of tiny aquatic creatures and feed on nutrients in the water.
Which brings up an analogy I found incredibly helpful in the research for making sense of this, because it's a very genetically fluid organism, right?
Really?
So think of the baseline V. cholerae as just a standard, perfectly safe car chassis. It rolls off the evolutionary assembly line completely harmless.
I like that analogy.
Right. It only becomes a public health threat if it gets some extreme aftermarket upgrades.
Specifically, 2 genetic upgrades. The sources highlight the CTXAB gene, which produces the actual cholera toxin, and the TCPA gene.
And the TCPA gene creates a hair-like attachment structure called a pilus, right?
Exactly. So without those 2 very specific aftermarket parts, the chassis is benign.
But here is the crazy part. The origin of those aftermarket upgrades is, it's one of the most astonishing discoveries in modern microbiology.
Yeah, because the bacteria didn't even invent those lethal genes.
Right. So in 1996, these researchers, Matthew Walder and John Michelanos, they discovered that the cholera toxin genes aren't native to the bacterium at all.
They were delivered by a virus.
A virus infecting a bacterium. Which just sounds like something out of a sci-fi movie.
It really does. It's this phenomenal mechanism called lysogenic conversion.
Break that down for us. What exactly is doing the infecting?
So there's this specific filamentous bacteriophage, which is just a fancy way of saying a virus that targets bacteria, and it's called CTX5.
Okay, CTX5.
Yeah. And this virus infects the harmless V. cholerae bacterium, and it literally integrates its own viral genome directly into the bacterial chromosome.
So the cholera toxin genes are basically carried along for the ride.
Exactly. The virus infects a harmless water bug and just permanently transforms it into a deadly human pathogen.
Wow. So the devastating disease we call cholera is actually the downstream symptom of a bacterium that's suffering from a viral infection itself.
That's a great way to put it. And that discovery totally shattered the old 19th century way of categorizing the disease.
Because back then, they viewed the cholera organism as a strict binary. You either had the germ or you didn't.
Right. But because virulence is actually a modular, transferable trait, those aftermarket upgrades.
Hmm.
That rigid 19th century definition is totally dead now.
It's obsolete.
Completely.
Yeah.
Today, microbiologists recognize over 200 different serogroups of E. cholerae.
And they classify those by the specific sugar coatings on their cell surfaces, right?
Yes, exactly. And almost all of those 200 serogroups are totally harmless.
But occasionally, one of those serogroups catches the right viral upgrades, and boom, you get an epidemic.
Yeah. And for over a century, epidemic cholera was almost exclusively blamed on just one toxigenic lineage.
The O1 serogroup.
Right, O1.
Yeah.
Seemed like the sole culprit. But then in 1992, in the Bay of Bengal, this completely new virulent strain just emerged out of nowhere.
That was O139, right?
Yep, O139. It had acquired the toxin genes and sparked a mass outbreak. So the definition of what causes epidemic cholera is constantly being redrawn by these genomic tools because the organism itself is a moving target.
Okay, so if this bacterium is such a genetically fluid shapeshifter, how on earth did 19th century scientists ever manage to pin it down in the first place?
Oh, it was messy.
I mean, it seems impossible given the tools they had back then.
Well, the historical origin story is fascinating. It involves this profound scientific coincidence right around the year 1854. You basically had two brilliant minds attacking the same invisible enemy but from entirely different angles.
So picture London in 1854. The Soho district is just being ravaged by a terrifying cholera outbreak. And this physician named John Snow starts mapping the deaths.
Literally going household by household.
Yeah, tracking the spread. And through this meticulous spatial mapping, he traces the entire cluster of deaths to a single contaminated water pump on Broad Street.
It's legendary in public health.
Totally. He convinces local officials to just remove the handle of the pump and the outbreak drops off. So Snow essentially invents modern epidemiology right there.
And he proves the disease is waterborne.
Exactly, and he does it without ever looking through a microscope to actually see the germ.
Right. But meanwhile, during that exact same year, you have an anatomist named Filippo Pacini working in Florence, Italy, during a totally separate outbreak.
And he takes a completely different approach.
He does. He actually examines the intestinal tissue of cholera victims under a microscope, and he observes enormous numbers of these comma-shaped organisms just vibrating under the lens.
Right, which is why he called it Vibrio.
Exactly. He correctly proposes that this living microorganism is the cause of the disease, names it, and publishes his findings.
Which is just, I mean, he had the microscopic assassin dead to rights. He saw it, named it, published it.
He did.
Yet the medical community completely ignored him for 3 decades. It is incredibly frustrating to read about his discovery just being brushed aside like that. Why was the evidence of his own eyes not enough?
Well, you have to consider the immense power of the medical consensus at the time. In 1854, the dominant paradigm was miasma theory.
Ah, right. Bad air.
Exactly. The greatest medical minds of the era firmly believed that epidemic diseases spread through foul, toxic air miasmas rising from filth and decaying organic matter.
Which, to be fair, probably intuitively made sense to them because impoverished areas smelled terrible and had the most disease.
Sure, the correlation was there. But Pacini's idea of a tiny, invisible germ living in the water, that directly contradicted a massive institutional consensus.
It's a classic case of an established paradigm just blinding people to new evidence. I mean, if the establishment is completely convinced the air is killing people, they aren't going to look in the water.
Exactly. And the shift only happened when someone with enough institutional weight challenged that paradigm.
Robert Koch?
Yes. The German physician Robert Koch, in 1883 and 1884, he led a government commission to investigate cholera in Egypt and then in Calcutta.
And he successfully isolated and cultured the organism, right?
He did. He called it the comma bacillus and aggressively argued that this specific waterborne germ was the sole cause of the disease.
But crucially, Koch wasn't an unknown anatomist in Italy like Pacini. Koch had already discovered the bacteria that causes tuberculosis.
He had absolute rock star status in the scientific community.
Right, which gave him the authority he needed to finally crush the miasma theory once and for all.
And this whole sequence of events is such a stark lesson in how scientific credit and memory operate. Koch had the fame, he had government backing, and he had the timing.
Yeah, for decades, the microbe was universally known as Koch's comma bacillus.
It took almost a full century for the microbiological community to go back, recognize Pacini's original 1854 publication, and officially rename the species Vibrio cholerae Pacini 1854.
Wow. Reputation and institutional leverage really shape the history books as much as the actual discoveries do.
They really do.
So now that we know what this bacterium is and, the battle to actually discover it, we need to look at what happens when human biology actually encounters it.
Because Koch proved it enters through the mouth via contaminated water, so you have to swallow this assassin.
Right. But the journey from a glass of water to the human gut is not an easy one for the bacteria, is it?
Not at all. It faces a brutal physiological gauntlet, primarily the human stomach.
Because it's so acidic.
Exactly.
Hmm.
V. cholerae is highly sensitive to acidic environments. Unlike an airborne respiratory disease where inhaling just a few viral particles can cause infection, cholera requires a massive oral infectious dose.
How massive are we talking?
Historically, estimates suggest you have to swallow anywhere from hundreds of millions to over 10 billion bacteria to get sick.
10 billion just to get sick.
Yeah, because the vast majority of those bacteria are utterly dissolved by the hydrochloric acid in your stomach.
Which explains a really counterintuitive risk factor mentioned in the epidemiological files. If you regularly take antacids, or if you've had gastric surgery, or suffer from a condition that lowers your stomach acid, your risk of contracting cholera spikes dramatically.
Because you've essentially lowered the drawbridge on your body's natural acid barrier.
Wow. And the acid barrier also explains the danger of eating undercooked, filter-feeding shellfish from endemic areas, right? Like oysters.
Oh, absolutely. Oysters and crustaceans filter massive amounts of water, which concentrates the bacteria in their tissues.
And if you eat them raw.
If you eat them raw, the dense protein of the food acts as a buffel against your stomach acid.
Yeah.
It basically protects the bacteria and delivers a massive concentrated dose straight into the alkaline environment to the small intestine.
Where they absolutely thrive.
Where they thrive, exactly.
Okay, so let's assume the bacteria survive that acid gauntlet and make it to the small intestine. This is where the microscopic hacking begins, right? Utilizing those aftermarket upgrades we talked about.
Yep. The first step is anchorage. The bacteria deploy that hair-like pilus to physically bind themselves to the intestinal wall.
Just to make sure they aren't washed away by the body's natural digestive flow.
Exactly. And once they're secured, they begin secreting the cholera toxin.
The mechanics of this toxin are fascinating. It's called an AB5 type toxin.
Right. Structurally, it looks like a ring of 5 B subunits surrounding a single active A subunit.
And the 5B subunits, they're responsible for targeting, right?
Yes. They bind to a very specific receptor on the surface of your intestinal cells. And once they lock on, they create a pathway to inject the single active A subunit directly inside the host cell.
And once that A subunit gets inside the cell, it basically acts as a molecular saboteur. It targets a regulatory G protein and just permanently locks it into the on position.
The cell essentially loses control of its own signaling.
Right.
Locking that G protein in the on state triggers this catastrophic biochemical chain reaction. It causes a massive sustained rise in a signaling molecule called cyclic AMP or CAMP inside the cell.
And that flood of CAMP forces open specific chloride channels in the cell membrane, right? The CFTR channels.
Yes. And interestingly, these are the exact same channels that are mutated and non-functional in patients with cystic fibrosis.
Oh, wow.
Yeah.
But the cholera toxin does the opposite.
Exactly. It blasts these channels wide open. So chloride ions just pour out of the cell into the gut lumen, and sodium and water rapidly follow through osmosis.
So the body's own cells are basically tricked into aggressively pumping their water directly into the intestines.
Precisely. And the clinical result of this cellular leak is absolutely devastating.
It produces what is medically documented as rice water stool. The sources describe it as diarrhea that is so voluminous and so completely drained of solid organic content that it literally looks like the cloudy, pale water you pour off after rinsing raw rice.
It's horrific. And the sheer scale of the fluid loss is hard to even comprehend. A previously healthy adult can lose 15 to 20 liters of fluid in a single day.
20 liters.
Yes. It's an extreme, rapid depletion of the body's entire fluid and electrolyte volume. Without intervention, the blood volume drops so low that the heart cannot pump it effectively.
So the patient can die from severe dehydration and hypovolemic shock in just a matter of hours.
Within hours of the first symptoms, yes.
But, looking at this from an evolutionary perspective raises a major philosophical question for me.
Okay, with that.
Well, if this bacterium naturally evolved to live in coastal waters, and human infection usually kills the host in a matter of hours, Isn't human infection basically a genetic dead end?
It seems like it, yeah.
So why did it evolve this devastating, highly specific toxin in the first place? I mean, destroying the host so rapidly seems entirely counterproductive.
That is actually one of the most compelling debates in modern microbiology. And the current evolutionary hypothesis offers a pretty surprising answer.
Which is,
The cholera toxin was likely never designed by evolution to harm humans at all.
Wait, really?
Yeah. Remember its natural habitat on the shells of tiny aquatic crustaceans, those copepods we mentioned.
Right.
The prevailing theory is that the toxin's real evolutionary function is to disrupt the microbiome and the biological biofilms of those aquatic organisms.
Oh, to help the bacteria compete for nutrients in the water.
Exactly. So the human tragedy is just an accident of biology.
Yep.
We swallow contaminated water, and the bacteria find themselves in an environment that just happens to perfectly and fatally trigger their genetic programming.
So the human intestine just serves as this highly effective, albeit accidental, amplification and dispersal chamber.
That's all it is to them. The massive fluid loss isn't a targeted attack to kill the human. It's simply the mechanical process that shoots billions of freshly multiplied bacteria back out into the water supply.
Where they can continue their natural life cycle.
Exactly.
An accidental amplification chamber, that is chilling. And when you are losing 20 liters of fluid a day, you are fighting a ticking clock. How does modern medicine even fight back against that kind of rapid fluid loss? You'd naturally assume that the response is hitting the patient with a massive dose of super antibiotics, right?
You would think so, but no. Antibiotics are frequently used, like doxycycline or azithromycin, but they are strictly an adjunct treatment.
Meaning they help, but they aren't the main cure.
Right. They can reduce the duration of the illness and the amount of bacteria shed, but they do not save the patient's life on their own.
Because the bacteria aren't invading deep tissues.
Exactly. They aren't attacking organs directly. They're just sitting on the surface of the gut, causing this catastrophic fluid loss. Death from cholera is purely death by dehydration.
So if you administer the strongest antibiotics in the world but fail to replace the lost fluids, the patient still dies.
100%.
Which brings us to a massive breakthrough in the late 1960s, culminating in what is often called the Bangladesh miracle.
Such an incredible story.
So, during the 1971 Bangladesh Liberation War, cholera was sweeping through these massively crowded refugee camps along the border with India. The medical camps were overwhelmed, and intravenous fluids were in desperately short supply.
They simply did not have the glass bottles and IV needles required to save everyone via an intravenous drip.
So medical staff desperately needed a way to rehydrate patients orally by simply having them drink fluids.
But the problem mechanically is that the cholera toxin has forced the gut's normal absorption pathways into reverse. It's aggressively pumping water out.
Right, so if a patient drinks plain water, it just passes right through the digestive tract without being absorbed at all.
Exactly. But researchers realized they could bypass those damaged pathways entirely. They developed a perfectly balanced oral rehydration therapy, or ORT.
And that's just a mix of specific ratios of glucose, sodium, and salts into clean water, right?
Yes. And by doing that, they found they could exploit a completely different active transport pathway in the gut wall, specifically the sodium-glucose cotransporter.
So the glucose actively drives the sodium across the cellular membrane, and the water just follows it back into the bloodstream.
Yes. Even while the cholera toxin is still firing away at the other channels.
It is an incredibly elegant physiological workaround. And the impact documented in the field was staggering. Physicians treating the refugee populations demonstrated that mortality rates, which routinely hovered around 30% or more for untreated cases,
Dropped dramatically.
yeah, they plummeted to well under 1% with the prompt administration of oral rehydration therapy.
From 30% to under 1%. It's amazing.
Just by giving patients a precise, cheap mixture of sugar, salt, and clean water. Public health historians constantly cite ORT as one of the most cost-effective medical interventions in all of human history.
It has saved tens of millions of lives from various diarrheal diseases.
Which is incredible. But the sheer simplicity of the cure highlights a really tragic reality of our modern world.
It does.
Yeah.
Because if the treatment is so accessible and inexpensive, it begs the question of why we are currently living through the 7th cholera pandemic.
Right. This current pandemic, driven by the L-Tor biotype, began in 1961 and continues to cause widespread outbreaks today. If the cure is basically sugar and salt, why is cholera still a global health emergency?
Fundamentally, cholera today is a disease of broken infrastructure. It acts as an immediate symptom of systemic societal collapse.
Like the devastating Yemen outbreak that began in 2016.
Exactly. Amid a tragic civil war, the country's water, sanitation, and healthcare infrastructure completely collapsed, and it quickly escalated into the largest cholera outbreak recorded in modern history.
With well over 1 million suspected cases.
Right. The medicine itself is simple, but if a population has no access to clean water to mix the ORT packets, or if they're entirely displaced by conflict, mortality inevitably skyrockets.
We also see how human movement and global infrastructure play a role in the 2010 outbreak in Haiti.
Oh, that's a landmark case study in modern epidemiology.
Following the devastating earthquake, Haiti experienced an explosive cholera outbreak, sickening hundreds of thousands and killing around 10,000 people. And what made this particularly shocking was that Haiti had not documented a single cholera case in roughly a century.
Right, so scientists sequenced the entire genome of the outbreak strain in Haiti and compared it against global databases.
And they discovered the strain was nearly identical to a specific South Asian strain of V. cholerae, didn't they?
Yes. Through rigorous genomic tracing, the source was linked to sanitation failures at a base housing United Nations peacekeeping troops.
Troops who had recently arrived from Nepal, a country where that specific strain of cholera is endemic.
Exactly.
The historical record shows that the United Nations actually initially resisted accepting responsibility for that. But an independent panel commissioned in 2011 concluded the evidence pointed strongly to the peacekeeping base as the source.
And it took until 2016 for the UN to issue a formal acknowledgement and apology alongside a response plan. This episode is now widely taught in epidemiology and international law courses because it demonstrates the profound power of genomic tracing to reconstruct the exact origin of an outbreak.
Providing undeniable scientific evidence, even when massive institutions are reluctant to accept the findings.
Right. And it proves that in a highly connected world, a microbe can travel thousands of miles inside an asymptomatic carrier because a large majority of infected people show mild or no symptoms and spark an epidemic the exact moment it encounters a vulnerable region with compromised sanitation.
It is a stark reminder that advanced medical technology isn't always the shield we assume it is. Our health outcomes with cholera track directly with access to the basics.
Clean water, functioning sewers, and basic oral rehydration salts.
Right. And while oral vaccines like Dukoral and Shunkle do exist and are used in emergency campaigns, the global demand during sudden crises repeatedly outstrips manufacturing capacity, forcing agonizing rationing decisions on the ground.
Exactly. So taking a step back, the journey of this organism is just remarkable. It really is. We start with a mostly harmless, comma-shaped microbe swimming in brackish coastal waters, feeding on copepod shells.
Then, through a viral hijacking by the CTX5 bacteriophage, it acquires the genetic code to produce a devastating toxin.
A toxin that likely evolved to disrupt aquatic biofilms, but accidentally turns the human gut into a massive amplification chamber.
It locks the body's cellular machinery in the on position, draining up to 20 liters of fluid a day. Yet it can be defeated by a perfectly balanced handful of sugar and salt.
It's wild. But before we conclude, there's one final eerie concept from the microbiology files that completely shifts how we view the intelligence of this organism.
Oh, what's that?
It's a mechanism called quorum sensing.
Quorum sensing here?
As these bacteria multiply by the billions inside a patient's gut, they aren't just blindly dividing. They actively communicate with each other using chemical signals.
Wait, they communicate?
Yes. They use specific autoinducer molecules called CAI1 and AI2. They're literally taking a census of their own population density.
Wow. But you would naturally assume that the larger the bacterial army gets, the more aggressively they would attack the host.
Right. But counterintuitively, they do the exact opposite.
Yeah.
Once the bacterial population inside the gut hits a critical density, they use this chemical quorum sensing to collectively turn their virulence genes down.
They turn them down.
Yes. It is an orchestrated collective exit strategy. The bacteria chemically agree that the human amplification chamber is full.
That is unbelievable.
They stop attaching to the intestinal wall, they shut down the toxin production, and they basically prepare to ride that catastrophic flood of fluid back out into the environment.
Perfectly primed to infect a new host or just return to the ocean.
Exactly, a strategic retreat.
So they chemically communicate to coordinate an exit. It completely redefines what we call a simple organism. Well, thanks for joining us for this deep dive into a truly mind-bending microbe. Stay curious, everyone.